Everyone, today in this video we'll discuss about antigen processing through MHC class 1 pathway. So as we know that antigen processing is an important or essential part for the removal of this antigen from our body. Antigens are the foreign particles and these foreign particles when enters in our body they induces a immune response. So these antigens can be a bacteria or a virus, anything. Okay, so these pathogenic bacteria or virus are very harmful for our body, and once they enters in our body they start showing their effect, which can be very detrimental. So these pathogenic bacteria or virus which are harmful for our body, our immune system needs to recognize them and eliminate them from our body. So in this antigen processing part, the adaptive immune system, which consists of T cells or B cells, plays a very important part in this adaptive immune response. So what happens here? The antigens that enters, which is usually a bacteria or a virus, they are processed or chopped off into small pieces, and they are presented together with MHC molecule. So I will make a video where we will discuss about this MHC molecules and what is this MHC molecule, why this system chooses this MHC molecule for the presentation of this antigen. MHC molecule serves a very important function here. MHC full form is major histocompatibility complex. So MHC molecules, they bind with the antigen, okay, and this MHC molecule has a function. They present this antigen to T cells and they have the ability to discriminate between self and non-self cells. Self means the cell that is present in our body, that is our body tissues or cells, and non-self cells means those that are the invader or the foreign ones. So MHC has the ability to distinguish between self. So our body chooses this particular system, this MHC, to bind with this antigen and then come over the cell surface of antigen presenting cell, which can be macrophage, dendritic cells, B cells. Okay, so these antigen presenting cells, they present this process antigen together with MHC molecules to the T cells that are cytotoxic T cells or the helper T cell that will involve the B cells on antibody mediated clearance. So all these antigen presenting cells plays a very important role in presenting this processed antigen together with MHC molecule on the cell surface, where they get recognized by this T cells, which is cytotoxic T cell, the CD8 and the CD4, that will involve the B cell process or antibody mediated clearance. Okay, so in this video we will discuss about this cytotoxic T cell mediated clearance. So as we have said that there are two different pathway. One takes place via MHC1. Okay, so the one that takes place via the MHC1 pathway and the other one that takes place via MHC2 pathway. So in this slide we will discuss about the endogenous antigen or MHC1. So the beauty of our immune system is that they have developed a mechanism for the clearance of both endogenous antigen as well as the exogenous antigen. So endogenous antigens are produced within our own body cells that has been infected by a virus. Okay, so our body has developed a way for the clearance of both kind of antigens. One that is endogenous, that are present inside the cell, and the other one that is exogenous, that comes from outside the cell. Their replication takes place outside the cell and they are exogenous. And the one that are present inside the cell, their replication takes place inside the cell This kind of antigen refers to endogenous antigen. So what happens? Suppose the replication of the translation of viral protein is taking place in the cytoplasm. Okay, so the clearance of protein that takes place in the cytosol is mainly mediated by proteasome complex. So suppose a ubiquitin tag has been given to this viral protein which is needed for clearance. So the proteasome complex will clear this viral protein, will chop off this viral protein into the small pieces, and these small pieces of protein, viral protein, will then needs to get clear from the cell, or they needs to get presented towards a T cell so that they will ultimately gets clear off from the body. So for this viral protein to get clear off from the cell, it first needs to get enter inside the endoplasmic reticulum. The reason that it needs to get enter here is the presence of MHC1 molecule inside this ER. So how does it enter this ER? This viral protein enters this ER via TAP1 protein. Okay, so this TAP1 protein facilitates the entry of this processed viral protein via the proteasome complex. So TAP1 protein function is, these are the transporters that are associated with antigen processing. So this antigen processing will enter via this transporter, that is TAP1. TAP1 interacts with tapacin as well as this ERP57 protein, and together all this protein comprises the protein loading complex. So what is this protein loading complex? Protein loading complex means they will load this protein, this processed protein, into this MHC1 groove, binding groove. Okay, so once this viral protein enters into this endoplasmic reticulum via this TAP1 protein, it depends on their size, whether this size is about 8 to 10 amino acid or more. If the size of this protein is more than 8 to 10 amino acid, they will undergo further cleavage via this endoplasmic reticulum associated proteases to bring them into a size that is almost of about 8 to 10 amino acid. Since bigger amino protein of since protein that consists of more than 10 or 12 amino acid will not be able to bind here. Okay, so it must needs to get chopped off into small fragments of almost about 8 to 10 amino acid. So beside this, there is another event take place, is the maturation of or the proper folding of this MHC1 molecule. So the proper folding of this MHC1 molecule is done with the help of two different protein. One is calnexin protein, that is denoted by CNX1. Calnexin protein plays a very important role in the folding of this nascent MHC1 to a mature form, and it also helps in the association of beta 2 domain via this MHC1 molecule. So we will discuss about the structure of MHC1, and I hope you guys know about the structure of MHC1 molecule and what does this MHC1 molecule comprises of. So MHC1 molecule, they basically consist of this alpha chain and this beta part. So this MHC1 class 1 molecules are heterodimers that consist of this alpha 1, alpha 2, alpha 3 chain. So if you look at this side of MHC1, it consists of alpha 1, alpha 2, and this three part, alpha 1, alpha 2, alpha 3, and it gets stabilized when it get interacts with this beta 2 globulin part. Okay, so this beta 2 microglobulin interacts with um, MHC1 molecule via this calnexin protein, and it gets stabilized. But when this binding site is empty, MHC1 molecules are not stable. So to its stability, to increase its stability, this another chaperones, this calreticulin, they bind this or they load this MHC1 complex into this protein loading complex. So when this MHC1 molecule interacts with this protein loading complex that comprises of this TAP1, tapacin and ERP57, then in that case they undergo a conformational change, and that conformational change makes them more stable and attains a configuration that has a higher affinity to bind this processed polypeptide. So let me recap again. MHC1 in this nascent form gets properly folded via two different protein. One is calnexin protein and other one is calreticulin. So calnexin protein helps in the interaction of this beta 2 microglobulin with the alpha 1, alpha 2 and alpha 3 chain of this MHC1 molecule and stabilizes it. But since it does not bind to its ligand or any protein in its binding cleft, they are unstable, and to make it more stable, calreticulin loaded this MHC1 empty MHC1 complex into this protein load peptide loading complex to make it more stable, because binding of this MHC1 molecule with this TAP1 tapacin as well as ERP57 induces a conformational change in this MHC1 molecule and also make them attain a configuration that is more favorable to bind this process peptide. So once this peptide binds to this, or once this peptide are loaded via this complex to MHC1 molecule, the affinity of this MHC molecule for tapacin gets reduced, and then moves towards Golgi, and from Golgi it then cell surface where CD8 T cells will come. T cells having this receptor will come and recognize this and do the clearance function. Okay, so in case of this endogenous antigen processing, the proteins are first chopped off into pieces in this cytosol via proteasome complex. This chopped off protein then enters into the endoplasmic reticulum via this TAP1 complex. This TAP1 comprises of protein loading complex together with tapacin as well as ERP57. Now, two chaperones rather play a very important role in the proper folding of this MHC1 molecule and its attachment to the protein loading complex. These are calnexin and calreticulin. So once this MHC molecule comes in contact with this protein loading complex, the processed protein or peptides are loaded into this binding cleft of MHC1, and once they bind, the structural change in this MHC1 molecule reduces its affinity towards this PLC complex and make them move towards the Golgi body. From Golgi they are exposed to the cell surface, and then the CD8 T cells come and recognize them, and further process are done. So this is what occur in this endogenous antigen processing pathway, and in the next lecture we will discuss about how the exogenous antigen processing takes place. So thank you for watching my video.